Fasciola hepatica, Wikipedia audio. Fasciola hepatica, also known as the common liver fluke or sheep liver fluke, is a parasitic trematode of the class Trematoda, phylum Platyhelminths. It infects the livers of various mammals, including humans. The disease caused by the fluke is called fasciolosis or fascioliasis, which is a type of helminthiasis and has been classified as a neglected tropical disease. Fasciolosis is currently classified as a plant/foodborne trematode infection, often acquired through eating the parasites Metacercariae and ceasted on plants. F. hepatica, which is distributed worldwide, has been known as an important parasite of sheep and cattle for decades and causes significant economic losses in these livestock species, up to £23 million in the UK alone. Because of its relatively large size and economic importance, it has been the subject of many scientific investigations and may be the best known of any trematode species. F. hepatica's closest relative is Fasciola gigantica. These two flukes are sister species. They share many morphological features and can mate with each other. Fasciola hepatica occurs in the liver of a definitive host and its life cycle is indirect. Definitive hosts of the fluke are cattle, sheep, and buffaloes. Wild ruminants and other mammals, including humans, can act as definitive hosts as well. The life cycle of F. hepatica goes through the intermediate host and several environmental larval stages. Intermediate hosts of F. hepatica are air-breathing freshwater snails from the family Limnaeidae. Although several limnaeid species susceptible to F. hepatica have been described, the parasite develops only in one or two major species on each continent. Galbatron catula is the main snail host in Europe, partly in Asia, Africa, and South America. Limnaea viator, L. neotropica, Pseudosuccinia columella, and L. cubensis are most common intermediate hosts in Central and South America. Several other limnaeid snails may be naturally or experimentally infected with F. hepatica, but their role in transmission of the fluke is low. The list of limnaeid snails that may serve as natural or experimental intermediate hosts of F. hepatica include: the Metacercariae are released from the freshwater snail as cercariae, and form cysts on various surfaces including aquatic vegetation. The mammalian host then eats this vegetation and can become infected. Humans can often acquire these infections through eating freshwater plants such as watercress. Inside the duodenum of the mammalian host, the Metacercariae are released from within their cysts. From the duodenum, they burrow through the lining of the intestine and into the peritoneal cavity. They then migrate through the intestines and liver, and into the bile ducts. Inside the bile ducts, they develop into an adult fluke. In humans, the time taken for F. hepatica to mature from Metacercariae into an adult fluke is roughly 3 to 4 months. The adult flukes can then produce up to 25,000 eggs per fluke per day. These eggs are passed out via stools and into fresh water. Once in fresh water, the eggs become embryonate, allowing them to hatch as myricidia, which then find a suitable intermediate snail host of the Limnaeidae family. Inside this snail, the myricidia develop into sporocysts, then to rediae, then to cercariae. The cercariae are released from the snail to form Metacercariae and the life cycle begins again. Life cycle. Fasciola hepatica is one of the largest flukes of the world, reaching a length of 30 mm and a width of 13 mm. It is leaf-shaped, pointed at the back, and wide in the front. The oral sucker is small but powerful and is located at the end of a cone-shaped projection at the anterior end. The acetabulum is a larger sucker than the oral sucker and is located at the anterior end. The outer surface of the fluke is called the tegument. This is composed of scleroprotein, and its primary function is to protect the fluke from the destructive digestive system of the host. It's also used for renewal of the surface plasma membrane and the active uptake of nutrients. On the surface of the tegument are also small spines. Initially, these spines are single-pointed, then, just prior to the fluke entering the bile ducts, they become multipoint. At the anterior end of the fluke, the spines have between 10 and 15 points, whereas at the posterior end, they have up to 30 points. The tegument is a syncytial epithelium. This means it is made from the fusion of many cells, each containing one nucleus, to produce a multinucleated cell membrane. In the case of F. hepatica, no nuclei are in the outer cytoplasm between the basal and apical membranes. Thus, this region is referred to as anucleate. Instead, the nuclei are found in the cell bodies, also known as tegumental cells. These connect to the outer cytoplasm via thin cytoplasmic strands. The tegumental cells contain the usual cytoplasmic organelles. The tegument plays a key role in the fluke's infection of the host. Studies have shown that certain parts of the tegument can actually suppress the immune response of the mammalian host. This means that the fluke is able to weaken the immune response, and increase its chances of a successful infection. A successful infection is needed for the fluke to have enough time to develop into an adult and continue its life cycle. Ostropepli allula, Ostropepli tomentosa, Ostropepli viridis, Fossaria bulamoides, Galba truncatula, Limnia cousini, Limnia cubensis, Limnia diaphana, Limnia humilis, Limnia neotropica, Limnia occulta, Limnia stagnalis, Limnia viatrix, Omphiscola glabra, Pseudosuccinia columella, Radix auricularia, Radix ligatus, Radix natalensis, Radix peregra, Radix rubiginosa, Stagnicola caprata, Stagnicola fuscus, Stagnicola palustris, Stagnicola turricula. The alimentary canal of F. hepatica has a single mouth which leads into the blind gut. It has no anus. The mouth is located within the anterior sucker on the ventral side of the fluke. This mouth leads to the pharynx, which is then followed by a narrow esophagus. The esophagus, which is lined with a thin layer of epithelial cells, then opens up into the large intestine. As no anus is present, the intestine branches, with each branch ending blindly near the posterior end of the body. Flukes migrate into smaller capillaries and bile ducts when feeding within the host. They use their mouth suckers to pull off and suck up food, bile, lymph, and tissue pieces from the walls of the bile ducts. F. hepatica relies on extracellular digestion which occurs within the intestine of the host. The waste materials are digested through the mouth. The non-waste matter is adsorbed back in through the tegument and the general surface of the fluke. The tegument facilitates this adsorption by containing many small folds to increase the surface area. F. hepatica has no respiratory organs. The adult flukes respire anaerobically. Glycogen taken from within the host is broken down by glycolysis to produce carbon dioxide and fatty acids. This process provides the fluke with energy. In contrast, the free-living myricydia stages of the parasite generally develop within oxygen-rich environments. The free-living stages of the parasite are thought to respire aerobically, to gain the most energy from their environment. F. hepatica's excretory system contains a network of tubules surrounding one main excretory canal. This canal leads to the excretory pore at the posterior end of the fluke. This main canal branches into 4 sections within the dorsal and ventral regions of the body. The role of F. hepatica's excretory system is excretion and osmoregulation. Each tubule within the excretory system is connected to a flame cell, otherwise known as protonephridia. These cells are modified parenchyme cells. In F. hepatica, their role is to perform excretion, but more importantly, osmoregulatory functions. Flame cells are therefore primarily used to remove excess water. The nerve system of F. hepatica consists of a pair of nerve ganglia, each one is located on either side of the esophagus. Around the esophagus is a nerve ring, which connects the 2 nerve ganglia together. The nerves stem from this ring, reaching the posterior end of the body. At the posterior end, one pair of nerves becomes thicker than the others, these are known as the lateral nerve cords. From these lateral nerve cords, the other nerves branch. Sensory organs are absent from F. hepatica. F. hepatica adult flukes are hermaphrodite, each contains both male and female reproductive organs. The male and female reproductive organs open up into the same chamber within the body, which is called the genital atrium. The genital atrium is an ectodermal sac which opens up to the outside of the fluke via a genital pore. The testes are formed of 2 branched tubules, these are located in the middle and posterior regions of the body. From the epithelium lining of the tubules, sperm is produced. The sperm then passes into the vas deferens and then into the seminal vesicle. From the seminal vesicle projects the ejaculatory duct, and this opens into the genital atrium, and many prostate glands surround this opening. The right side of the anterior testis has a branched, tubular ovary. From here, a short oviduct passes to the vitelline duct. This duct connects, via a junction, the ovaries, the uterus, and the yolk reservoir. From this junction, the uterus opens into the genital atrium, this opening is surrounded by Melas glands. In some flukes, the terminal end of the uterus is strengthened with muscles and spines. F. hepatica reproduces both sexually, via the hermaphrodite adult flukes, and asexually. The Myristidia can reproduce asexually within the intermediate snail host. Currently, F. hepatica has one of the widest geographical spread of any parasitic and vector-borne disease. Originating in Europe, it has expanded to colonize over 50 countries, covering all continents except Antarctica. In contrast, F. gigantica is generally considered more geographically restricted to the tropical regions of Africa, Asia, and the Middle East, with some overlap between the 2 species. Morphology and anatomy. Climate affects both F. hepatica and its definitive host, the snail. For example, the development of F. hepatica myristidia and larvae, and the reproduction of Galba truncatula, require a temperature range of 10 to 25 degrees Celsius. In addition, they both require high levels of moisture in the air, as both are at risk of desiccation. Due to this, the prevalence, along with the intensity of infection, of F. hepatica is primarily dependent on rainfall levels and temperature. F. hepatica's tegument protects it from the enzymes of the host's stomach, whilst still allowing water to pass through. Free-swimming larvae have cilia and the cercariae have a flagellum-like tail to help them swim through the aquatic environment and also allow them to reach the plants on which they form a cyst. To attach within the host, F. hepatica has oral suckers and body spines. Their pharynges also help them to suck onto the tissues within the body, particularly within the bile ducts. The adult fluke's respiration is anaerobic. This is ideal, as no oxygen is available in the liver. F. hepatica is adapted to produce a large number of eggs, which increases its chances of survival, as many eggs are destroyed on release into the environment. Also, F. hepatica is hermaphrodite, thus all flukes can produce eggs, increasing the number of offspring produced by the population. The genome for F. hepatica was published in 2015. At 1.3 GB, its genome is one of the largest known pathogen genomes. The genome contains many polymorphisms, and this represents the potential for the fluke to evolve and rapidly adapt to changes in the environment, such as host availability and drug or vaccine interventions. For more information on the epidemiology, see the disease page. Fasciolosis. Infection begins when cyst-covered aquatic vegetation is eaten or when water containing metacercariae is drunk. In the United Kingdom, F. hepatica frequently causes disease in ruminants, most commonly between March and December. Humans become infected by eating watercress or by drinking emollient, a Peruvian drink that uses drops of watercress juice. Cattle and sheep are infected when they consume the infectious stage of the parasite from low-lying, marshy pasture. Human infections have been reported from more than 75 countries around the world. In Asia and Africa, people are infected both by F. hepatica and F. gigantica, whereas human fasciolosis is caused only by F. hepatica in South and Central America and Europe. Tegument. Digestive system. The presence of F. hepatica can interfere with the detection of bovine tuberculosis in cattle. Cattle co-infected with F. hepatica, compared to those infected with M. bovis alone, react weakly to the single intradermal comparative cervical tuberculin test. Therefore, an infection from F. hepatica can make it difficult to detect bovine tuberculosis. This is, of course, a major problem in the farming industry. Respiratory system. Excretory system. Nervous system and sensory organs. Reproductive system. Prevalence. Both F. hepatica and F. gigantica can cause fasciolosis. Human symptoms vary depending on whether the disease is chronic or acute. During the acute phase, the immature worms begin penetrating the gut, causing symptoms of fever, nausea, swollen liver, skin rashes, and extreme abdominal pain. The chronic phase occurs when the worms mature in the bile duct, and can cause symptoms of intermittent pain, jaundice, and anemia. In cattle and sheep, classic signs of fasciolosis include persistent diarrhea, chronic weight loss, anemia, and reduced milk production. Some remain asymptomatic. F. hepatica can cause sudden death in both sheep and cattle, due to internal hemorrhaging and liver damage. Fasciolosis is an important cause of both production and economic losses in the dairy and meat industries. Over the years, the prevalence has increased and it is likely to continue increasing in the future. Livestock are often treated with flukicides, which are chemicals toxic to flukes. The two chemicals used are triclobendazole and bithionol. Ivermectin, which is widely used for many helminthic parasites, has low effectivity against F. hepatica, as does praziquantel. For humans, the type of control depends on the setting. One important method is through the strict control over the growth and sales of edible water plants such as watercress. This is particularly important in highly endemic areas. Some farms are irrigated with polluted water, hence, vegetables farmed from such land should be thoroughly washed and cooked before being eaten. The best way to prevent fasciolosis is by reducing the limnaeid snail population or separating livestock from areas with these snails. These two methods are not always the most practical, so control by treating the herd before they are potentially infected is commonly practiced. Parasitic adaptations. A diagnosis may be made by finding yellow-brown eggs in the stool. They are indistinguishable from the eggs of Fascioloides magna, although the eggs of F. magna are very rarely passed in sheep, goats, or cattle. If a patient has eaten infected liver, and the eggs pass through the body and out via the feces, a false positive result to the test can occur. Daily examination during a liver-free diet will unmask this false diagnosis. An enzyme-linked immunosorbent assay test is the diagnostic test of choice. ELISA is available commercially and can detect anti-hepatica antibodies in serum and milk. New tests intended for use on fecal samples are being developed. Using ELISA is more specific than using a Western blot or ARC2 immunodiffusion. Proteases secreted by F. hepatica have been used experimentally in immunizing antigens. Epidemiology. Fasciolosis. Diagnosis.